Chapter Principles of Inheritance and Variation. Mendelian laws consist of law of dominance, law of segregation, law of independent assortment. Monohybrid cross. Monohybrid phenotypic ratio 3:1. Genotypic ratio 1:2:1. Test cross and back cross. Test cross means the crossing of F1 progeny with the recessive parent. Test cross ratio 1:1. Back cross means the crossing of F1 progeny with any one of the parent. Example of incomplete dominance. F1 progeny shows a character in between two parents. Dominant gene is incompletely dominant over recessive gene. Genotypic and phenotypic ratio of incomplete dominance is 1:2:1. Multiple allelism and codominance. Alleles from the parent 1 IA, alleles from the parent 2 IA, genotype of offspring IA, blood group A. Alleles from the parent 1 IB, alleles from the parent 2 IA, genotype of the offspring IB, blood type AB. Alleles from the parent 1I, parent 2I, genotype II, blood type O, etc. Multiple allelism. The involvement of more than two alleles for a single trait. Example, human blood group. Codominance. Two dominant alleles are equally dominant and express at the same time AB blood group. Dihybrid phenotypic ratio 9:3:3:1. Sex determination. XX, XY type, Drosophila and human, male heterogamy. XX, X0 type, insect, grasshopper, male heterogamy. ZW, ZZ type, birds, female heterogamy. Pedigree analysis. Symbols. Genetic disorders. Mendelian disorders, hemophilia, bleeder's disease or royal disease. Clotting of blood is delayed. Sex-linked recessive trait, sickle cell anemia, autosomal recessive trait. Substitution of glutamic acid by valine at the 6th position of beta globin chain. Picture of normal hemoglobin chain and sickle cell anemic hemoglobin chain. 2. Phenylketonuria, inborn error in metabolism. Next, chromosomal disorders. Down syndrome, 55 autosome plus XX or XY. 47, 21 trisomy. Short stature, mentally retarded. Klinefelter syndrome, 44 autosome plus XXY. 47, sterile male. Gynecomastia. 3. Turner syndrome, 44 autosome plus X0 equal 45, sterile female, rudimentary ovary. Next chapter, molecular basis of inheritance. A. Nucleoside. Nucleoside is nitrogen base plus pentose sugar. Nucleotide, nitrogen base plus pentose sugar plus phosphate group. Bond present between nucleotides, phosphodiester bond. Central dogma. It is proposed by Francis Crick. The double helical model of DNA proposed by Watson and Crick. Double helical model of DNA contains two polynucleotide chain. Nitrogen bases ATGC sugar deoxyribose. Hydrogen bond connect nitrogen bases between A and T. 2 hydrogen bond between G and C. 3 hydrogen bond. Phosphodiester bond connect sugar and phosphate. Heterochromatin and euchromatin. Heterochromatin densely packed, euchromatin loosely packed. Heterochromatin it stains dark, euchromatin stains light. Heterochromatin it is transcriptionally inactive, euchromatin active. Difference between DNA and RNA. DNA double stranded, RNA single stranded. DNA deoxyribose sugar, ribose sugar. DNA nitrogen bases are A, T, G, C. RNA nitrogen bases AUGC. DNA purine and pyrimidines are present in equal amount. RNA purines or pyrimidines are not present in equal amount. DNA replication, semi-conservative mode of replication proposed by Watson and Crick. Structure of replication fork. Transcription unit. Properties of genetic code. Genetic code is triplet, commaless, unambiguous, degenerate, and universal. AUG is the start codon. It codes for methionine. Stop codon UAA, UAG, UGA. Template strand 3' TAC, GTA, CGTA, CGTA, CG 5'. Coding strand 5' ATG, CAT, GCAT, GCATG, C 3'. mRNA strand is given here. Cistron, it is a segment of DNA coding for a polypeptide. Lac operon. DNA fingerprinting. It is developed by Alec Jeffrey. Steps of DNA fingerprinting: isolation of DNA, digestion of DNA by restriction endonuclease, separation of DNA fragments by gel electrophoresis, transferring of DNA fragments into nitrocellulose paper blotting, hybridization using labeled VNTR probe, detection of hybridized DNA fragments by autoradiography. Application of DNA fingerprinting used in forensic science to solve the problems of paternity, rape, murder, etc. Used to diagnose genetic disorders.